The following two talks is about cancer, is make our life better, to prevent cancer, to treat cancer. So life expansion and make our life better. So these two parts fits well. Is this really a global meeting, you think? Fertility, women, now we cancer research guys are put together. I mean, and from all around the world. I like this. Typically, we go to an ACR meeting in the country. There's everybody talking about the cancer. Now here, it's talk different. Plus, the food is very good. So I'm talking about particularly one kinase. I want to focus on very small, and I try to focus on with this arrow gamma here. And I don't have a good pointer if I use my finger because of coffee is shaking. I will move. I can make a mess here, but I will try to. So I'm talking about PCD gamma. I want to say number one, why I'm talking about PCD gamma. Why PCD gamma is important? Okay, I want to particularly make a point. PCD gamma is important for GI cancer. Two types of GI cancer I'm going to talk about today: colon cancer and pancreatic cancer. So pancreatic cancer, my colleague already have me with some background information. I don't need to go detail. I just want to narrowly focus on PCD gamma. So, and what is PCD gamma? What is this guy doing? So let's see these slides. And this is I don't have a point here, but anyway. So this is the MAPK pathway. If you graduate maybe from the last 5 years, you learn this in grade school. Otherwise, maybe you read already this in literature. Okay. So the three major MAPK pathway: ERK pathway, JNK pathway, PCD pathway. And PCD pathway is inflammation pathway. Inflammation is important. I can say every cancer involves inflammation. I mean, also you can say every disease involved inflammation. But how inflammation works, we don't know. Okay, this is a global picture like this. So this is a paper we published 23 years ago. So something like this, my wife and I'm always talking about past. So this is history class. This is a paper we published 23 years ago, almost last century. Okay, and what we found in this paper, actually, this is my first R1. It's memorable. So rats make three MAPK pathway: ERK, JNK, PCD pathway activation. What we found in this JBC paper, we block an activation of PCD pathway by rats. Okay. See, I cannot go back. And I don't have a pointer. Apologize for that. Yeah, if somebody has a pointer, would it be helpful? Yeah, because my hand is shaking. If I use my finger, and then all slides will be mess. This is a PowerPoint. Cannot do it, right? Can do it. Don't have a pointer with this.
Full screen.
Not in presentation mode. Apologize, waste of your time, but I want to make it better. Apologize.
Pointer. There is a pointer. Okay.
Oh, thank you very much. Oh, that's very helpful. Okay, this is just history, and you don't need to remember this. I just want to share a link. This is maybe this JBC paper first showing PCD link to RAS oncogene. So RAS is number one oncogene, important for many cancers, particularly pancreatic cancer. So that is this 2000. This is JBC paper showing PCD alpha is linked to RAS signaling by negative feedback. And then when we look at P38, okay, there's a 4 gene on different chromosomes. We want to see how important this different P38 in cancer. So this is a logical question everybody will go. Okay, if you have 4 brothers and sisters, we want to see how each of you are doing. This is rational where we will go. Okay, what we found from our last 20 years work from 2 institutions where I worked, we found P38 gamma is doing different job than P38 alpha. Actually, opposite job. What we found before P38 alpha is tumor suppressor. Actually, last 20 years, our work found P38 gamma is oncogenic, is required for cancer development, particularly in colon cancer, pancreatic cancer, and breast cancer. And also, second part from this slide is we have established P38 alpha inhibitor, SB compound. Everyone knows SB, anti-inflammatory, but not many people know what is P38 gamma inhibitor. We don't have official P38 gamma inhibitor yet, so anti-inflammatory study. So this is a busy slide. For my grant application 20 years ago, I will share with you briefly. You don't need to pay attention to this. This is the complicated slides. So what we show here, let me talk briefly. What we found, P38 gamma RAS make P38 gamma RNA expression go higher, make P38 gamma protein expression higher. And this elevation P38 gamma protein, P38 gamma gene is important because you knock down P38 gamma, you block RAS cancer growth in cell culture. If you don't have RAS mutation, you don't see these results. So this is important. We show this in colon cancer cells. We show this in colon cancer xenograft. This is xenograft. This is cell culture. This is xenograft. This is cell culture study. So this means P38 gamma is important in colon cancer. So we do next. If you argue, if I want to say P38 gamma is important, we want to ask the next question: knock out this gene P38 gamma, what happens? And this is my knockout study in inflammation. So this side, we shoot these cells. This side is we treat with 5 days with DSS. DSS is inflammation chemicals. You put into bottled water to feed the mice every day. After 5 days, so mice will develop colonitis. During this colonitis, you see this is controlled normal, and we have a DSS treatment. That's inflammation. Inflammation increase P38 gamma protein expression, not alpha. Okay. And this is gamma, so this is higher than this side. If you see alpha, you don't see how to see this. You see phosphorus higher. Okay, this is HE staining, and this you can look at this specifically. See this more when you take up colon tissue, you do IP experiment. Look at phospho-P38 gamma. You can confirm this. Means what this means is that in mice, inflammation. Specifically, phospho-P38 gamma, not alpha. Okay, indicating specific phosphorylation may be some role for P38 gamma in inflammation-induced colon cancer. So this is the oncogenesis study. We put a DSS and then treat the mice with LM. LM is carcinogen. So carcinogen plus DSS make colon cancer. If we have a control mice like this mice, you see this, maybe this first time for you to see what is colon cancer looks like in mice colon. Okay, this is a beautiful colon cancer in mouse. If you knock at the P38 gamma on this side, substantial decrease. Okay, knock P38 gamma out, and specifically, or let me say pathologically, we knock out P38 gamma, not whole body knockout. It's this intestinal epithelial cell specific condition knockout. this is term you don't need to remember, but indicating we is calling many anatomy structure. So typically cancer develop from epithelial cells. So we knock PCD gamma out specifically from epithelial compartment. We block colon cancer development. So this means it's relevant to the disease. Not only gene knockout, inhibitor. I mentioned PCD gamma is no good inhibitor, but we found that this a student in the lab, they find a compound called PFD. PFD is anti-inflammatory agent, is using now in European to treat many colon inflammation for bowel disease, inflammation disease. So it's FDA approved agent using to treat patients with colon inflammation. But we are the one recognize this is important for colon cancer. So we put this in this mice. We see similar things. If you treat these mice with PFD, so compared to the control group, so tumor develop much less. Molecular level, we can block many genes, down regulations by this compound, PCD gamma inhibitor. What this means genetically, pharmacologically, we can hit PCD gamma, and then we can treat colon cancer, or we can prevent colon cancer development. We can say like this, although many people, especially review, they don't like my comments. And this one is we treat these mice in PCD gamma knockout mice. We don't see that phenomena. That means PCD gamma is important for this chemical, for the inhibitor to play a role. If we don't have gamma there, and you don't see effect. And this is the molecular analysis. We did I think we identify Wnt signalings important in colon cancer, particular beta-catenin for this type of cancer. And this is the molecule you see knock out the mice, the Wnt target genes, PCD gamma are gone. Okay, confirm this in mice and human colon cancer specimens. And so this is the molecular mechanism. We found inflammation stimulate and PCD gamma bind to beta-catenin, and also we identify PCD gamma can phosphorylate beta-catenin on specific residue on serine 605. It is a complicated story. I don't just want to make simple. Serine 605. And this phosphorylation is important for PCD gamma to stabilize beta-catenin. So the conclusion for the colon cancer stuff is PCD gamma is required for inflammation-induced mouse colon cancer, which may be involved in human pancreatic cancer because we have tissue that support this kind of correlation. Okay. Now let me move to a second cancer and pancreatic cancer. Second cancer is the history is like this. This red and in signaling field, almost every pancreatic cancer has a RAS mutation. Okay. And how this RAS mutation causes pancreatic cancer, we don't know. Another phenomenon, this is NAM, Wamber effect. It's a German name, Wamber. This name comes from Nobel Prize about 100 years ago. So what Wamber found this phenomena is, he found all cancer cells compared to normal cells. So cancer cells generate ATP in different way than normal cells. This is huge. Even this after 100 years research, I mean, that's why he got Nobel Prize. And this cancer cell like different living than normal cells, specifically get ATP. And then this is almost in every case of KRAS mutated cells. So KRAS mutant cells will use this type of effect because RAS can make this one-way effect. And how KRAS make this? And nobody knows. But some spontaneous research have shown RAS can make more PFK3 glycolytic protein, make other protein. So indicating RAS can make GLUT1 glucose transporter. These two glycolytic metabolites, enzymes. These two enzymes contribute to glycolysis, make glycolysis similar way in cancer than different way in cancer than in normal cell. But the background is how RAS make this happen. Nobody knows. We want to ask if PCD gamma play a role in this process. So easy one experiment we did is we knock down PCD gamma protein here, and we see the separation of cell growth in two separate KRAS mutated pancreatic cancer cells. Detected in cell culture, maybe it's working. And then we use PCD gamma and the PCD delta inhibitor. So this is a PCD alpha inhibitor, not much effect. The gamma effect to cell line, few effect. So in cell culture, genetically, pharmacologically, maybe we got something. And to confirm this, we have a panel of pancreatic cancer cell line, normal cells, and normal cell transfected with RAS, and we use this PCD gamma inhibitors, different combination. But we found only PCD gamma inhibitors suppress cancer growth, but normal pancreas cells growth. So this means something specific with PCD gamma in KRAS. So, okay, they link together. That's a special link. And PCD gamma with KRAS. And then we want to go detail about the mechanism. We did this proteomic analysis. Oh, thank you, sir.
Thanks.
So what we did, we took KRAS transfected pancreas cells, and then we analyzed PCD gamma intact which protein we want to identify PCD gamma specific interacting protein in a KRAS dependent manner. No KRAS, no interaction. Okay. So this is this is PCD gamma transfected. This is control transfected. And then we put on PCD gamma, analyze the protein to see which protein binds PCD gamma. So we want to see, this is the KRAS mutant cells. We want to see PCD gamma interacting with which guy. And then we found PCD gamma is here, and the gene name of PCD gamma is called MAPK. This protein is the PFK3, indicating this is the glycolytic enzyme may be important for PCD gamma activity. So we confirm this in Western blot. You can see this KRAS cell, we have RAS, and we have PFK3 here, and we have PCD gamma here. If we use cells transfected with PCD gamma alone, it can show similar things. PCD gamma here, and this PFK3 here, and GLUT2 here, not GLUT1. So GLUT1 we did not identify this assay, but GLUT2 may be in the pathway by different mechanisms. Okay. Now from some of these results, if we knock down KRAS, both all the molecules down. If we knock down PCD gamma, more or less similar things, indicating so PCD gamma is important for KRAS in the cell line. This is a little bit more complicated in mechanism. I just want to go brief. And then we identify PCD gamma can phosphorylate PFK3 on this specific residue. And these are all slides we want to show together is PCD gamma can phosphorylate PFK3 on this residue. And consequently, PCD gamma can make this protein more stable. And could be important in biology. This is all we try to show. Next, please. Oh, you make my life easier. Thank you. So this is PCD gamma that in and pancreas inflammation. This is we generate knockout mice, condition knockout, and this is different evidence knockout, and this is in evidence for inflammation. So here we use thoroughly to induce. So in colon cancer study, we use DSS to induce colon inflammation. In pancreas, we use thoroughly to induce. Okay. So in PCD gamma knockout, so inflammation decrease, histology and quantitative, and this pro-inflammatory cytokine decrease. So it means PCD gamma is pro-inflammatory. And this different molecule conform and these conclusions. Next, please. Okay, this is phenotype. Is this important for cancer? Yes. This is genetic evidence, KPC, 1 level knockout, and 2 levels double knockout. If knockout PCD gamma is genes, the mice can survive more 100 days. So a mouse, how long mice will survive typically? More than 6 months, okay. But this is knockout one gene and survive 100 days more, and this is very substantial. This part we do like it. That's why we can make a kind of research paper. So this is evidence we have knockout. This is molecular pathology. We have knockout showing different things. Next, please. Oh, this is some kind of data show like just like clinic may be used for pancreatic cancer diagnosis. This is evidence showing you knock out PCD gamma, you block glycolysis pathway. Okay. Glucose uptake, enzyme activity, lactate secretion, ECA, OCA, and we have a tumor xenograph, and we have this CT scanning. Okay, so overall, so you knock down PCD gamma, this all them together suggests PCD gamma is important in glycolysis. Knock out PCD gamma, you block glycolysis, you block mice cancer growth and development. This a little bit of further detail. So the point is here. Overall, I want to analyze the two molecules. One is GLUT2, one is PFK3. And this slide, two slides showing is one point is I want to demonstrate PCD gamma require or collaborate with these two molecules because these two molecules make glycolytic pathway. Okay, these two molecules, GLUT2 and PFK3, and they are both molecules signaling downstream of PCD gamma. If PCD gamma is there and you regulate these two molecules, you have an effect. Biology. If you knock out PCD gamma, you don't have this effect. And this effect happening through phosphorylation because you phosphomutant, we have phenotype, for example. You make more, put PCD gamma more into the cells, you have more cell growth. If it is mutant, and this is phosphorylation residue knock out mutant or mutated with mutant, you don't have this stimulating animal. In morphology, in tumor growth, indicating PCD gamma phosphorylation PFK3 is important for pancreatic cancer growth in this model. This slide, I want to demonstrate not only genetic, pharmacologically, I'm a pharmacologist. I want to prove everything in pharmacology because potentially, if I have a good inhibitor, so this inhibitor potentially could go to clinical trial. This is my overall goal. This white up, yes. It might not correct as the knockout, showing some effect. In PFD, this is survival much longer for PFD treatment. If you knock out the PCD gamma in the mice, no effect. So the evidence for the effect. This is KPC mice in primary pancreatic cancer. If we use xenograft, coming from this, so this tumor, mice tumor, nice is you can take these tumor cells, put into the culture, and then you transfect with different plasmid. and then you can put it back into the mice to test the phenotype. So here is, if we look at the PFD therapy, if you have a PCD gamma here, show effect. If you knock out PCD gamma and xenogram, no effect. I just want to say here, and this inhibitor, pirfenidone, PFD, is working through PCD gamma, both in the primary tumor and xenogram. This is a patient sample analysis. This is a postdoc who did this who collected sample in China, Pharma. I just mentioned to my colleague a while ago. She's now in Canton now, she's working. She did this collecting analysis in Canton. She has access to patient sample. I had the difficulty to get sample from my lab in USA. So what she found is, so all these 4 samples, they're higher in patient pancreatic cancer tissue. This is all 4 of them. So it means important for cleaning up pancreatic cancer. Oh, finally I'm here. So this is the previous or currently lab member contribute work what I talk mostly from Pharma for cancer research paper, and another guy is Mi Yi, and he's first author for the colon cancer study, oncology paper. And this is my collaborator. This is the funding support this study. Thank you very much for your time.